now, on to the next two companies in our competition and their judge, Orly Ben-Herzel, VP of Value Creation at Migal and CEO of Galileo Migal Tech Transfer. Please welcome the first company, Orly Savion, CEO of EcoPhage.
Hi, my name is Orly Savion. I'm the CEO of EcoPhage, a company that directs nature resources to control plant bacterial diseases. Our company was established by the Bayer Trendlines Innovation Fund and was built around technology developed by Professor Rotem Sorek from the Weizmann Institute of Science, a world leader for bacterial phage interaction. Our multidisciplinary team gathered to answer a major need. With 20 to 30% global yield loss due to more than 200 bacterial diseases. Some of them have no solution available at all, and the ones that do suffer from low efficiency, increasing bacterial resistance, toxicity, and limitation by regulation. The best place to look at is nature. Phages are the natural enemies of bacteria. These are viruses that specifically attack target bacteria, self-multiplying within their hosts, safe to the user and to the environment, biodegraded, leaving no residues at their surrounding. However, there's a technological challenge. Bacteria and phage diversity is very high. It is difficult to identify and to select the right phages, creating an effective and sustainable solution. EcoPhage combines nature untapped biodiversity with the cutting-edge science and technology from the Weizmann Institute of Science, ones that was already validated by a pharmaceutical company, both creating a platform for tailor-made phage solution to bacterial crop diseases. Our products and platform consist on 3 layers. First, the market-ready product, standard care for disease-specific prevention and treatment. And the second, customized products, on-the-spot solution for outbreaks, creating the third layer on our company's full vision, global data hub and AI analysis for bacterial diseases. A platform for outbreaks prediction and next-generation solution, combining climate, disease, crop, geolocation, and phage correlation to give the farmer an immediate and long-term solution, helping the environment by improving soil health, leading to increased yield, and giving the company a unique positioning for disease prediction, prevention, and treatment. Our achievement includes the funding by the IAA and funding by the Trendlines Bayer Innovation Fund due to successful milestones. The platform was already validated at lab experiments, plant assays, and greenhouse experiments. We have proof of concept for 2 indications with up to 80% efficiency and 100% global coverage of bacteria. Our products roadmap consists on the development of the 2 existing products, reaching sales at the end of 2024, adding to expansion of the pipeline for additional indications. We are currently launching our Round A funding to reach field trial and EPA filing for the first 2 indications, real-time condition validation for the extra additional indications as well, and achieving a proof of concept for computational system and database. Thank you for listening. We are EcoPhage.
Thank you, Orly, for this amazing presentation. You seem to have made some very exciting achievement in those 2 years or so. My first question is about safety challenges. You mentioned specificity, and I'm mainly curious about if there is any risk at all, and if so, how do you plan to overcome aspects of specificity and the prevention of overgrowth or drift of phages into non-target bacterial domains or populations? And in this kind of drift could be creating some kind of resistances, mutations, lysogenes, and DNA particle transitions. So yeah, this is my main question.
Okay, so first of all, thank you. Actually, phages are considered very safe to the surrounding. First of all, they are highly selective. They only target their specific bacteria. They're not harming humans, other organisms, including insects, and also other beneficial bacteria. So we only use phages that are lytic and not lysogens that can actually incorporate to the DNA. These phages terminate the bacteria and leave no residues at all. Once the target bacteria is terminated, the phages are actually dissolved to the surrounding and keeping it safe. By the way, the phages come from nature originally, so we don't create any difference in balance during time. And as for regulation, we are not the first one to regulate phage-based solutions. There's already EPA approval for a product, which was very permissive, by the way, including some substantial waivers that relates to toxicity. So basically, considering the other possibilities that the farmer has, this is the safer solution that he can think of. So we're very confident in that.
Okay, thank you for that. Second question is about delivery and application. So, especially now during the era of COVID and its vaccine and public discussion around it, the issue of different types of nucleic acids and the resistance to changing environmental conditions is at the forefront of research among scientists and arouses a lot of curiosity and awareness among the population. How do you make phages work in challenging environments, such as fields or greenhouses, and if it is applicable in large-scale operation?
So the good thing about phages, we collect them naturally, and nature actually did the selection for us. So we only choose phages that are already very stable, but we do, however, further characterize our phages to make sure that these are stable in conditions that relates to storage and also to field conditions. This is part of what we do. As for the scale-up possibilities, so the good thing about phages is that their manufacturer is our target bacteria. So we use fermentation in order to produce the phages. And this is a very scalable process. We actually even started doing the scalability of this procedure, and it's very cost-effective. So again, phage is a very good selection to use as a natural solution.
It sounds like it. Okay, so thank you very much, and good luck and all the best.
Thank you.
Thank you, Orly. This is the time to cast your vote.